Welcome to Mr. Chalk's Revision Tips. In this video, we will look at
plant cloning, adult cell cloning, and embryo transfer. So in biology, cloning is the process of producing similar populations of genetically identical individuals that occur in nature when organisms such as bacteria or insects, plants or animals reproduce asexually. Plant cloning is easier than using seeds to generate new plants because they duplicate, or the duplication of desired genes is rapid, and the grower knows the type and quality of plant that he would produce. Embryo transplants is a process of splitting apart cells from a developing embryo before they can become specialized. These can then be grown into individual transplant embryos and transplanted into host mothers. And tissue culture is another new technique that's been used for cloning plants. So here only a few plant cells are actually needed. These can then be added to a growth medium, and hormones will help them develop into a new plant. So we're explaining what is meant by a clone. So a clone is something that is genetically identical. The main advantages of cloning plants is that it's quick, and cloning is an example of asexual reproduction. So if we're looking at cloning plants, we'll start off with tissue culture. So one way of cloning plants is by tissue culture, which works not by having cuttings, but by having tiny pieces from the parent plant. So sterile agar jelly with plant hormones and lots of nutrients is needed. So this makes tissue culture more expensive and difficult than taking just plant cuttings. And cuttings is the simplest way to go and clone a plant. All you need to do is take a cutting off the parent plant, put it in some rooting powder, for instance, then go and replant it, and it will grow into an entire new clone of that parent plant. A developing embryo in an embryo transplant is removed from a pregnant animal at an early stage before the embryo cells have had time to become specialized. So the cells are separated and grown for a while in a laboratory and then transplanted into host mothers. So the way that this happens is that a developing embryo is removed from a pregnant animal at an early stage. The cells are separated and grown for a while. So when the offspring are born, because these new embryos will be implanted into surrogate mothers, so when the offspring are born, they will be genetically identical to each other. And they will not be identical to their host mothers because they contain different genetic information than the surrogate mother that they were transplanted into. So if we're looking at what stage of development does the egg need to be split up, it's when it's early on, so it's an embryo and the cells haven't differentiated. So once divided, what the embryo is inserted into, that would be surrogate mothers. Fusion cell cloning involves replacing the nucleus of an unfertilised egg with the nucleus from a different cell. The replacement nucleus can come from an embryo, but if it comes from an adult cell, it is called adult cell cloning, right? So this was how Dolly the sheep was produced. So the first thing is that we take a cell from the sheep that we want to clone. So an egg cell is taken from a donor sheep. The nucleus is extracted from the donor egg and got rid of. The cell from the first sheep that we want to clone is inserted into the empty egg. The cells are given a small electrical shock to go and fuse them together. The egg then develops into an embryo, and the embryo is implanted into a surrogate until it develops into a clone of the first sheep, and it will have the same genes as that first sheep that we wanted to clone. So scientists use an egg from a living mouse and genetic material from a brain cell of a frozen mouse. So describe how the process of adult cell cloning could be used to clone the frozen mouse. So the key things that we need to talk about is that we're removing the nucleus from the egg, we're inserting the genetic material, the chromosomes, from the frozen mass, and we need to go and give it a little electric shock in order to go and fuse the cells together and get them to divide, and then go and insert them into the womb or a uterus of a surrogate mother.
Thanks for watching.